All right, thank you all for coming. It is my pleasure to introduce Edward Narayan. He is a self-described champion of animal welfare and conservation physiology. He did his PhD at the University of South Pacific, which I have visited. It's in Suva, yeah?
Yeah.
Which is a very beautiful place in Fiji with great access to outdoors. Studied frogs?
Yeah.
You're gonna tell us about it?
Yep.
Cool. He did postdocs.
Phobia. I have to say frog phobia, please. There will be pictures of frogs and videos.
Okay, cool. He did postdocs in New Zealand, Australia, India, and Canada.
Saskatchewan.
Oh, cool. And then he returned to Australia in 2010 with Griffith before eventually moving here. And now he is based at UQ at Gatton in SAFS, not the vet school, right?
No, that's why I'm moving around.
You were in the vet school?
No,
Okay.
I am affiliated
Okay, cool.
with Coffee and SVS as well.
So
I teach into the vet tech program.
okay.
Yeah.
What is that?
Bachelor of Veterinary Technology.
Oh, yeah, vet tech, got it.
All the vet nurses suture the skin and all. Yeah.
Cool. So he has a wide variety of interests around animal welfare, and he's gonna be telling that his story. I think a few different case studies today
Thank you.
from
Yeah.
his different research. So thank you for coming.
Thank you, everyone. And really great to see all of the colleagues joining us in person and also online. So thank you very much. I'm also here with a few of my students from Gatton who are coming to join us. So please feel homely. I'd like to acknowledge the traditional owners and their custodianships on the land on which we meet. Pay my respect to the ancestors, descendants, and with the continuous cultural and spiritual connections to the country. I recognize their valuable contributions to Australia and the global society. So as we've just heard about my career trajectory, we've got a few students, I believe, in the crowd. So this is where I was, and then moved around and have been at UQ for a bit over 5 years now. There has been a lot of footsteps and footprints for the work that I have gathered in the space of conservation physiology and animal welfare science. And so there is a lot of people whom I have been lucky enough to get to know and learn from. My professor, Mark Hero and Frank Moliana. I'm sorry, this one is a bit weak, but they have all helped me out. You might see that there's students as well who have been the champions. It's not just me. I'm not the champion. They are the ones who try to. So it's the stress lab. And so what the stress lab is basically is that we are trying in our group to understand about animals. So I have a lab where we can actually stochast reproductive and stress profiles of vertebrates. We've just had Yefi submitting a PhD thesis looking at the largemouth bass. So she went to China and studied so all the way from wildlife, agricultural animals, pets. We've had some students looking at the emotions of horses that are used for equine therapy, as well as dogs that are actually used for walk and to reduce stress in humans. So we are doing all sorts of research. There's a lot of animals that we haven't been able to tap into, but this is the beauty of, for me as a researcher, starting off from frogs, we are now actually able to learn. And I think the learnings that I am picking up every day, it's giving me a lot of things to work on. And the latest one, I think from here, is that I've just been offered got accepted for a book deal with Elsevier to write on stressing animals. So that's a huge commitment for me for the next 2 years. So conservation physiology is actually understanding the physiological response of animals to human alterations of the environment. We know Professor Craig Franklin. He was my mentor. He was my referee for my job here. So I always am highly indebted to Craig. The conservation physiology team. How many of you have heard of this? Please raise your hands. Brilliant. So we've got a few of us over here. So we try to look at the causative mechanisms of the underlying under the skin, how we can actually assess animal and their response to environmental change, and then make some contributions to the discussions and all the way from the ground level to policy actions and policy making. And you'll see some of the ways that we've been trying to do that as well. I will briefly mention about climate change. We all know what it is. It's basically the effect of humans contributing to the aggressive nature of how the atmospheric temperature has become ever more aggravated, and it's actually leading to macro and macro scale changes to our environment. And especially coming from Fiji and Pacific Islands, I'm feeling very vulnerable with what will happen to our communities. And I'm going back to Fiji in 3 weeks, and I'm trying to engage with the Nature Fiji to see if we can actually reignite some of the conservation discussions around how we can save some of these endemic species on islands, because time is against, we're running against time. So being lucky, some of the hard hitters in this game. So you can see that we've been trying to see how we can use physiology to be part of the conversation around predicting the global warming and also then mitigating, and so physiology is an important part of this. So this is where I was for almost 4 years doing my PhD. I took this picture on a flight to Vanuatu to meet my brother, who's a dentist on this big island, and it's been like removing teeth from all Fijians out there. And I said, oh, I've seen this island before. And I said, no, Edward, that's where I was. And so I quickly took the snapshot. You can see there's two halves. This is not working. I should just use my hand. There's two halves to the island where if it's really emotional for me because there's a lot of stuff going on over here. So you can see if it's high tide, you can go straight to the village. There's around 50 people on the island, so it's 60 hectares. And that small island over there, or the little topu, that's a traditional spot for funerals and things. It's very, very sacred. So this island is actually encompassing ground frogs. So for me as a 20-year-old, my supervisor, Craig Morley, who is a well-known ecologist from New Zealand, he said, Edward, I will throw you to this island, you're gonna go and catch frogs. I was into textbooks and getting A+ and whatever, I didn't know what I'm signing up for, but that's what my life was, to go and collect frog pee. And you'll know why I was doing that. But the frog is actually all over the island. And so when I landed the island, I discovered that it's a mecca of ground frogs. It's just unbelievable. And we'll have a chat about that. Hopefully you'll have lots of questions to ask me. So this is where we are. This is Fiji, Vanuatu, Vichi. This is where I was born. That little circle over there. Had my whole life on this little village with my parents, and then studied on Viva Island. It's very emotional for me to then come from Fiji to Australia and be here for the last 15 years or so. So how we can actually understand the ecology of some of the species of which virtually nothing is known. We are running against time. So I had two supervisors. One was a hardcore physiologist from India, Professor Christie, and then I had a frog ecologist or a biodiversity conservationist, Craig Morley. I was sandwiched in the middle as a PhD student to go and discover the biology of this unknown species. There was some records about the natural history of the ground frogs, but virtually not much was known. So Creighton said, Edward, go and cut up some frogs. We submitted the ethics application, got knocked back. You can't cut ground frogs, mate, because it's IUC and endangered species. So then turned the tide, and luckily at that time in New Zealand, they were trying to develop conservation physiology tools for the Archie's frog. So the Archie's frog is a highly threatened species in New Zealand, and so they were trying to develop a conservation physiology tool with Jen Germano, who's doing the PhD the same time as me in New Zealand, and I'm here in Fiji. And so we went into this expedition to collect frog urine. So it was very funny, a bit of a race as well. But the important thing here was that can we actually sequester the hormones and understand the ecology of these animals? Why we are trying to do that? What does it tell us? So basically you can see the connection between the brain and the behavior of the animals. I would say the behavior is secondary. There is already some sort of a physiological change that is happening inside the animal's body, which would then tune the animal's behavioral response. It may not always be the case. Sometimes you will have behavioral response in the absence of physiological change because this is energetically demanding. You have to use ATP to be able to then have that burst of energetic response for the muscles to perform its activity, because this would be like if the snake is about to catch the frog, he can jump away from that threat. So it's not a bad thing if you are secreting cortisol, but you will notice that through long time period, it's this scale of turning on and turning off of the stress response, which works around this concept of negative feedback, which is basically to maintain homeostasis. But as you will see, my research started off into studying frogs and also other animals in zoo settings and in very mild environments, where you can see that the turn on and the turn off of the response is very much clear. You can notice, you're moving koalas from one place to another, or in the zoo you're handling them. They will show a response in the turn off, but then I can see now with some of the work of my other team members, we are seeing that the turning off is not happening anymore, and so that's where we are now zoning into as well. Some of the work that Harsh is doing as well for his PhD. This is the fun of being on the island. These are not my kids. My 9-year-old saw this picture and was like, "Dad, what's all that? Why are you hiding these photos?" No, this is the fun of being on the island. Highly, I would say I'm in the right place. I feel like I'm at home here, and it's really amazing that that's Bow Island, where the first military sort of like the chiefly war happened between Tongans and Fijians for the rights of their land. And Viva Island is very much important because Bible was translated to the Fijian language on this island. So it's a very complex environment because one of the landscapes is where there's a pond, and Craig and I wanted to shut up that pond, but the priest used to bath in that pond. So it's actually a very complex place to work in if you were to do some conservation work, because we wanted to remove the cane-toads on the island as well. So I took all the frog pee to New Zealand, to Frank Moliana, who was at the Ecogene lab. So this is the New Zealand where I took all the frog pee and got stopped at the airport and all the humor of that. But then this is where we actually managed to discover for the first time, as you do in your PhD, that you can measure stress responses or reproductive and stress hormones from the same cocktail of urine sample of the frogs. So the beauty of this is that you would just go in. I remember collecting the samples. I didn't know if I'll measure anything in that. And I was like, 2 a.m., 3 a.m. in the morning with my head torch, catching frogs, collecting this sample that looks like just water. I was like, what the heck, would there be anything in this? But I had to believe in my professors because back in USP, there wasn't much resources available at the time to actually do any validation work. So I basically took a huge risk taking all the raw samples with me to New Zealand, but it actually worked. There's another story around that, but things did not work. Like they had to change the milky water and all other things. Lots of fun of PhD, the failures of PhD, but eventually it did work. And then that opened up a beauty of looking at the ecology of the animal and understanding what the physiology is doing. So some really basic, this is basic biology, it's not rocket science. Looking at how the testosterone profiles vary in the ground frogs with the Fijian wet season. If you go to Fiji during Christmas, it will be really, really wet. You must have caught in some of the cyclones in Fiji at that time. I remember my uncle and aunts used to come from Canada and they caught up in the cyclones, and we were little kids. And you can see the progesterone profile, the estrogen. So this is all animal welfare friendly. We are actually opening up the whole world of the biology of this animal non-invasively, which is really, really cool, I think. But the other thing was that what about the developmental biology? So this is a process known as candling, where you can actually put the frog, turn off your head torch, look at the eggs. So my job was to measure the eggs every month and then come up with a chart that would tell me that the female is actually gravid, she is at this stage of vitellogenesis and this stage not. This is the first time that I discovered the eggs of ground frogs. It was amazing. Fiji One News, like ABC, came on our doorsteps. Craig said, Edward, go and save these frogs and release them on another island. I said, okay. Ketan said, no, Edward, you look at the development biology and then do some other physiological, look at how it's affected by temperature. We can do a altitudinal gradient kind of a study looking at how temperature affects these frogs. What would happen? I said, no, mate, I have to finish my PhD. I didn't know what to do then. But basically what the end point was that these guys, that's what it looks like. Just take a little sip of water while you immerse in the picture I took. The good thing about these guys is that they actually have direct development. So they don't give birth to a tadpole. A baby frog comes out in 29 days. They have some really interesting synergies with crabs, with ants. The frog's jelly is actually covered with antimicrobial properties. There could be 10 PhD projects. I was talking to Sebastian before, and it's amazing how much more work can be done on this island. And that's, I think, where we need to look at as well. It's a really cool field site for students. So with Mark Hero, Jean-Marc Hero, he's at Griffith University, and Craig Morley and Catherine, the two, musketeers over here. And Mark was at Griffith University, so he was my PhD examiner. Then I did my postdoc at Griffith. He was fascinated by this stuff. He said, Edward, come and join me. He helped me to publish this paper. Not amazing microscopy, but we can actually see the whole developmental biology with some basic light microscopy in Fiji at that time. To see the actual development of the blood vessels, the vertebral column. They have a little bit of an egg for 5 days, egg yolk, on which they feed, and then it covers off afterwards. So they're really cool animals. Then basically what happens is that you have chronic stress, right? So this is all operating over here in the basic ecological scale, where you have this turning on and turning off, resting homeostasis, you have the predictive and responsive state. But then overall, if you remove the environment in some way, like pollutants or forest destruction, you end up actually affecting the negative feedback loop, and it leads to chronic stress. It's a very basic diagram over here, a bit sad. So I collaborated with Tim Jessop at Melbourne University, and we did a bit of a research on controlling the cane toads by setting up macro exclusion chambers. And that was my first ever big paper in functional ecology. So I was very proud of that. But basically what it demonstrated was that the reproductive output of the ground frogs would be affected by the fear inflicted by the cane toads on them. It's amazing, I'm seeing this research being cited similar to my other research from other landscapes, like with fire ants or with invasive lizards, between native reptiles or native snakes. All over the board, there are some really interesting synergies with how an invasive species can really double down on the ecological fitness of an endemic species. So we still have cane toads on this island, and my dream is to actually come up with some strategies where we can remove cane toads. People on the island are doing agriculture. They are having slush burning of the landscape. And I think there needs to be more that scientists and the government can do together to actually help the villages to be better equipped with looking after the ground frogs. They have high respect for the ground frogs. This is their totem. This is their. This is like similar to, like the wedge-tailed eagle or something like or the tigers in India or the elephants. Like this is a huge thing for them, and so it's going through a lot of problems. So then when I came to Griffith, there was an opportunity to go to India. And with the help of the Australia-India Early Career Fellowship through the Australian Academy of Science, I met Professor Grama Purohit, who was based at the University of Pune. That's where Harsh is from. So I met Harsh through there. And this is their laboratory. Beautiful people, beautiful students. Their laboratory, they're studying the ecology of some of the really rare endangered frogs in India at that time. And so I took my lab with me with the kids on a plane to India, help set up the project for them. And Amruta Joshi, actually, this was our field site in Matheran, which is actually in Pune. And so you can see that the male frog is guarding the eggs, the nuptial pads, and the beautiful landscape actually. And so we studied the frogs, and we then discovered Amruta Joshi, she finished her PhD a few years ago, and we were able to actually correlate the breeding ecology, calling behavior to testosterone profiles in the urine sample. How you collect frog from a tiny frog like this, how you collect urine from a tiny frog like this. There is a lot of technique involved. And so, yes, it was rudimentary when I was a PhD student, throw a cup, ground frog would just be on everywhere, But these guys actually are very fast. So you have to minimize their stress. And there was some optimization of the techniques with the work done by Phil Barn at the Wollongong captive breeding facility for corroboree frogs. And so we, with Amy Sila, Phil Barn, and the team, we actually have got some papers out there which actually talks about how you have microtubes that can actually help in sequestering enough urine that would be able to detect the hormones. So Mark, hero, he is a hero, my hero, and so he was fascinated by frog biology, and so he discovered that the Litoria wilcoxi would change color. He used to go hiking or cycling or whatever on the Springbrook, up the mountain, and that's Vito, and that's John Shuka, that's Mark sitting over there. I think I'm taking the picture. Vito arrived from Brazil that night, dragged him with his suitcase from the airport to the field site. And we actually looked at the color change behavior in the Litoria wilcoxi. Christina did a PhD, and we could actually see the relationship between corticosterone and testosterone with the color change behavior of the frog. So I don't have a video in here, but it was really cool because the frog would be brown, and then in 1 minute it would become bright yellow. So what's the reason of becoming detectable to my predators? Is breeding so important, or is it a trade-off of some other? So you can look up Christina's PhD work. This is something that's existing in reptiles and snakes as well, and it could also be an indicator of stress in animals. So then I teamed up with colleagues in the US. Caitlin Gabo, I have a high respect for Caitlin. She's been doing a lot of work in salamanders, waterborne hormones, and so we can now, and with Drew Davies and Zachary Forsberg, Zachary finished his PhD on Ellen, artificial light at night, and how that affects frogs. We wrote this paper. The aim was to actually, like somebody connected to me wanting to study the Lake Titicaca frog, which was, and so I just want to pick up my bag, stop the teaching, go and study all these frogs in different parts. So that's my dream. I don't think I will be able to achieve it. I need the students to do that for me. The connection is there, right? And so I think for 5 minutes, I'll talk a little bit about koalas. If you're okay, Jim? Yeah. So a little bit of a breather, immerse in that picture. I take a bit of water. So, this will be a short story, right? So basically with koalas, Mark said, Edward, you can actually measure the same stress markers in koalas and bilbies. So we've got papers on bilbies, koalas, wombats, tigers, and all, here the big connection with colleagues at Dreamworld. But then when I went to New South Wales, I actually started connecting with the koala rescue groups, because when you look at that, negative feedback loop, these guys are at the war zone of what's happening to them in terms of, habitat change and all that. So I'll cut the story short. Negative feedback loop is buggered up. You can see over here, cortisol then becomes a threat to the animal's longevity and survival. And this is basically some of the landscape. This is from South Australia, one of our field sites. Well, it is a field site. It's just a study site where koalas are found. And so these are some of the koalas that would be occurring in this landscape. So they are really much dispersed all over the board. You know what we have here in Brisbane with our colleagues doing amazing research on koalas here as well. So I basically asked the question, what is that? The International Fund for Animal Welfare said, Edward, why don't you look at this area? Well, before that, what we discovered was that koalas are actually coming up in cities. So
new research suggests koalas rescued from rural areas are more stressed than those from urban centres. It's believed to be because the native animals from more populated areas are accustomed to having people around. This koala, Joey, is having a final checkup. She's got her bling on. She'll soon be released back into the wild. It's one of hundreds treated at the Port Macquarie Koala Hospital every year.
Apart from disease, which is the main reason for admission, we see koalas hit by cars, unfortunately attacked by dogs.
When it comes to saving injured koalas, the animal's stress level can be a deciding factor.
So it can have a major impact on their treatment.
A new study from Western Sydney University has measured and compared stress levels in koalas rescued from urban areas with those found in rural settings.
We are trying to understand how they respond to environmental change.
Edward Narayan and his team used scat samples sent in from wildlife groups to detect stress hormones.
We found that the koalas actually that live in rural areas, in rural urban fringe, were the most stressed. What was really interesting to us was that koalas in urban areas were actually showing low signs of stress.
The researchers say the findings don't mean koalas in urban environments aren't stressed. It simply means they're less stressed and appear to have become better at coming into contact with humans. They believe it's evidence the animals can adapt to having us around, but they need to have protected habitats.
If we don't deal with stress with regards to our native wildlife, we will see more and more beautiful animals will end up becoming extinct.
Something no one wants to see. Jonathan Hare, ABC News, Sydney.
Yep, so what I did was I had a lot of data on that acute state of stress response from the work I did in healthy animals in Greenwald, Currumbin, along the Gold Coast, looking at Southport area. There were some healthy animals, right? And in Ballina, this is Ballina, so this is the Northern Rivers, really interesting landscape where we've got koalas actually now are cross-cutting. So I took my bag and went into this landscape. It's horrific. The people were looking freakishly stressed out. Like you could hear the thing that goes inside the water to build bridges would shake your house. Basically, you can't have a cup of tea. And so what they did was they created a connection, I think from Ballina towards Byron Bay area or something like that. I'm about to go there to have a look at what this actually is. And that was our field site. So basically what I did was run a marathon of all the stress data that I had on koalas. And this went through scientific review. And I had some really good people who know the field really well, who read my manuscript. And they actually, rather than just dumping it, because it's not an ecologist's fun thing where you have lots of data or you could do fun things. I'd love to do fun things with ecological modeling. I think I'm not an ecological modeler, but I think I was able to demonstrate how the stress levels are different in regards to different stressors. So like acute stress, dog attack, like that would not kick in your stress response. Like you would not measure that immediately, but the state of the environment, land clearance was significant. So that was a big one, and I'm really interested in doing more work in that space as well. How we can minimize stress in burn victims by working with the rescuers and the carers to minimize stress in these animals. And you'd see koalas flat on the ground. They do drink water, And chlamydia is a big one as well. So, we know chlamydia is a massive stressor for koalas. So, that was something that attracted a lot of attention. And then I discovered that I didn't know about this, but my work was presented at the parliamentary inquiry. So that was really important for us to then see that in some way our work was actually being presented as part of the community of practice that we have got to then advocate for this our animals. And so that was just a threw this one last night just to, I know we are walking around the policy and all these things in here, the Center for Biodiversity and Conservation. So I think you guys will be happy with me. I don't know. Maybe not. So yeah, that's the story with the koalas, guys. We got together, there were 13 authors on this paper. Joe is actually a second-year student, Renee Hausch, the stress lab team. And we actually wrote a review on how stress is affecting animals around the world. And I'd say that's where, I'm wrapping it up. You're most welcome to, I don't know if you're on X or not, but yeah, I'm really keen on doing 2 new projects. Looking for students. Ground frog work, that's a mecca of ecology. Like if you are keen to go in a rugged landscape, lovely, beautiful, and study biology of animals, it's not just ground frogs, there's snakes, lizards. I'm keen and I need support. And so I think support in terms of more team members who are believing in doing a positive change in the Pacific Islands. And then the koala, of course, is part of the discussion. So with that, thanking everyone. Thank you again to all the colleagues over here and online. I'll leave you with a little video about Joey on Magnetic Island munching on. So she was rescued. Her mom died and was rescued. So thank you very much.
All right, are there some questions for Edward?
Do you find with your research, because you have to gather your samples from excretion, it kind of affects the photograph later? So I know it would be pretty difficult to gather from, say, blood samples when you're in hospital and you're not inducing stress.
Yeah, so that's a really good question. For every species that you are trying to develop the assays, you need to understand the excretory lag time, right? And so we do some basic laboratory and physiological validations to then understand what's the window between the actual absolute baseline to when your acute stress response happens. Yeah.
Oh, okay. Did you find out how long it actually takes to excrete the stress hormones?
Frogs?
Yes, yeah.
Thanks for asking a question about frogs.
Well, yeah, the ground,
Bless you.
the ground frogs, yeah.
I love it. Yeah, that was my life to actually with Ketan, he's a digestive physiologist. So my supervisor, PhD advisor, and so we actually did HCG and ACTH challenge and discovered that the stress response was around 2 to 3 days. It was very short and mild, and so that was really good because I had to run and catch the frog and get the cup ready. Bang, he's done his job. Bag him in the Ziploc bag, give it to my field assistant, and so that stress. We then had some buckets out because John Cochran, who was my advisor from Messi, he's a penguin stress physiologist. He studied penguins, so we did the acute stress response. So we kept frogs in a bucket overnight, and then saw the actual secretion of the hormones throughout the night as well. So yeah, does that answer your question?
Yes, so then you can take that into account.
Yeah, so
not
for
you doing, you can
No,
take
sorry,
that.
yeah, so if for any new animal that you're trying to work on, you need to do, but it's very hard to do a biological validation on some charismatic animals or really rare animals like tigers. So we relied on the veterinary health checks. And mind you, the tigers still didn't like the needle. Why? They showed a response, which was good for us because we ticked that box, because that's important. So I think the next talk would be Harsh when he's talking about the different assay techniques, because he's gone really into my PhD student who's not here today. He's gone right deep into the technicalities of measuring hormones. But there's thousands of papers on hormonal measurements in all sorts of animals, right? And so I'd say, yeah, 1,000, yeah. And so there is absolute numbers around what the baseline levels are. And then you can actually measure not just in saliva or feces, but hair, and look at long-term responses as well. Yes.
Mine's not a bit of a different question, I would say. When the side, I think there's been a bit of a conversation on the social side of conservation of bringing wildlife to the table when we are making decisions. And when you start talking about the physiological responses, that's something that came up in my mind. Maybe that's a way of giving wildlife a voice. When we are making.
Beautiful question. Thank you for asking.
So yeah, I would like to hear your
Beautiful
thoughts.
question, and I think that's why we are doing all this, because we are all part of a family and we have got techniques that actually, has something to talk about what the animals are trying to tell us, because they have been here far longer than we have been here. And so our koalas, our frogs, our butterflies, our bees, and all these animals. And so stress seems just by chance is one part of the story. And so if you see animals are really sick and injured or are really showing signs of stress, that's actually a sign that there could be some problems for humans as well. So it's always easier to sell the human stress story, because if it stresses me, then I'm really, agitated. But I think there are community members, like we had, the foxes that were in the Brisbane area. There were some new people who didn't know much about the flying foxes, and they were trying to hey, just remove all of these guys, but then we try to understand how to live with animals. It's not easy, but I think more education, more community understanding, and scientists actually condensing their research in a way that is communicable, like you can be communicated in the pub or in an everyday language. I think that's where we need to be able to then reach out and reach in as well. So that's a bi-directional, so that we can make positive change. Yeah. It's not easy. You guys know more than me. Yes.
For animals displaying chronic stress levels and obviously hormonal stress, what kind of techniques can you implement to reverse that or lower the hormonal stress signals?
Yeah, I'm looking at Zhengi because she's studying the black bears in Vietnam. Zhengi, do you want to answer this question? Or say a few words while I don't talk much? Sorry to throw you out like that, but I know you can answer it.
We're still working on the different levels of cortisol. The animals show after the chronic stress and the acute stress. And I think in the level of physiology, the HPA axis, the negative effect, the negatives will eventually show up in the result of cortisol level. So we're still trying to understand, especially for my project, we are working with bears, and those levels of cortisol still need more analysis, and we should combine the behavior and other factors into account, and we combine this data, those behavior recordings, with the measurement result of the results to finally figure out this.
Beautifully said, and I think what Zhengi also, so we can see that there's plasticity, there's flexibility, and this is actually work stemming from the biomedical literature, the work of late Professor Bruce McEwen, who, lucky enough, I don't know how he followed me, he became my friend on LinkedIn. He's the father of stress biology, right? And so, and Sapolsky, why giraffes don't get gastric ulcer, his book, that's all actually telling us that animals actually have a bit of a flexibility to be able to cope with the stress, but we humans always get agitated by every single thing that is not in our comfort zone. So there is plasticity, and we've seen then I'd love to be talking to Animals Asia and maybe with the vet school, how we can look at dendrites, and they actually rebranch. It's like water the plants which are dying and they rebranch, and so animals behave in the same way. Thank you.
I've got to run, so thank you. But let's chat about community education engagement stuff.
Cheers. Thank you.
Ross, yeah.
It might come in your dream. I had dreams about frogs actually, I'm not mistaken. Everyone, you're talking about frogs, catching frogs in your dream, like what the heck?
We could do a study on that as well. Everyone's welcome.
Yes. It's not, yeah, it's a real thing that happened to me. Anyone else has any questions or thoughts? Hopefully it was all making sense.
All right, well, thank you very much. We know where to find you online. So let's give one more round of applause.